Okay, class, so now we're on part two of today's lecture, and we're going to continue on with looking at some of the biochemistry of proteins. So right before the break, we were just looking at sort of active sites, but there's also plenty of protein-protein interactions that occur within a cell. And so you can have different interactions occurring via sort of different sort of folds within the protein. You can have sort of a surface and a string come together. We actually saw the helix bundles, and so you can have coiled coils that occur between two helices, and you can also have protein-protein interactions that occur between two surfaces. And you can also get heterologous assembly of large macromolecules, right? It's just not, two proteins coming together. And so, these are sort of crude diagrams of, how things interact. This one's not so crude. It actually occurs, but, you can sort of think of this surface and string coming together almost in the same way that an antibody would recognize an antigen, and if you had a protein-protein interaction where two large globular proteins come together and their sort of surfaces touch, you can think of the surface. So don't get too hung up in the way these things look. It's just important to know that large protein macromolecules can come together to do different things, and we'll see this all along and even later today. Okay, so here's an antibody, and this is really sort of a great molecule for understanding protein-protein interactions, because antibodies bind antigens, and I'm sure you guys have heard a lot, especially with the COVID-19 pandemic, about antibody-antigen interactions, right? So there's treatments for COVID-19, which include Regeneron's antibody cocktail and Eli Lilly's antibody cocktail. There's also convalescent plasmid, and it's this antibody that sort of is responsible for neutralizing the spike protein on the virus to prevent it from being infectious. Okay, it also targets it for destruction by the immune system. But just a little note on antibodies, because they'll come up throughout the course of the term. It's one of these things I really want you guys to know and understand how antibodies work. Antibodies have a heavy chain and a light chain, and they're actually a tetramer, so there's two heavy chains. These are identical, and then there are also two light chains. There's disulfides within the various domains, and then there's disulfide bridges that connect the two halves of the antibody together, and also disulfides that connect the light chain to the heavy chain. Here's the antibody fold. This happens to be just the light chain, and so here you have one domain, and it has a disulfide, and then you have your variable domain, and it's these loops out here. These are complementary determining regions in the antibody that actually help with antigen binding. And so I sort of try to explain to people that you can sort of think about an antibody binding an antigen as almost like a hot dog, which would be the antigen, and the bun being sort of the variable domain of the antibody, right? So the hot dog is going to fit in the hot dog bun, much the same way that you're going to have this surface binding this string, right? This is a hot dog in a hot dog bun. And that's roughly how antibodies work. But the antibodies actually, will bind to the antigen in their variable region here, and this has just been sort of a great model to study protein-protein interactions. Which leads us to the next topic, which is equilibrium dissociation. Okay, so you can have a molecule A, and molecule A, once it's made, can sort of randomly associate with other molecules like B, C, and D. And basically the number of non-covalent interactions that occur between A and whatever other molecule will determine basically how tightly that molecule is associated with each other, right? And so here you're going to have a good surface-surface interaction between A and D, and not so good surface-surface interactions between B and C. And so these don't typically form, or if they do, they don't last long, and the majority of A is just going to always be associated with D. Okay, and so biochemists use equilibrium dissociation to basically define how tightly things bind. And so in this instance, you have dissociation where you have A and B together, and when they dissociate, they become A plus B. Okay, and so the dissociation rate is the dissociation rate constant times the concentration of A and B, right? How often is that going to come apart? Association is just the opposite, where you have A and B coming together to form AB. And so the association rate is equal to the association rate constant times the concentration of A times the concentration of B. And so when you have equilibrium, and that means basically the association is equal to the dissociation, you get K on AB is equal to K off AB. Okay, so then you can find your equilibrium constant. Okay, so different molecules are going to have different equilibrium constants. So antibodies tend to fall around 10 to the 7th. The strongest biological interaction I think that's known is biotin with streptavidin. That's sort of something the graduate students might come across at some point in time. And then here's just the free energy difference between them as they're sort of happening with, depending on how tight or how large the equilibrium constant is. All right. No need to necessarily memorize this, just understand equilibrium dissociation. And if you sort of understand it in the big picture here, that the more non-covalent interactions, basically the higher, the higher affinity and the higher equilibrium constant that's going to come out. That's what, I really want you to know. At some point in your life, you guys may need to actually calculate the equilibrium constant or the equilibrium dissociation constant. I've had to do it once in my life. It was like 24 years ago. So it's just not something you come across every day. Important for biochemists, important for defining how strong something binds, but as a general rule, it's not something we do every day. Okay, so here's some common enzyme families, and we've seen a couple already. You have hydrolases. These are basically enzymes that sort of lead to hydrolytic cleavage, and there's subcategories of those like nucleases, which cleave up nucleic acid, proteases, which break down proteins. In the opposite, you have synthetases. These actually are in anabolic processes. They typically cause condensation reactions. Isomerases, we already saw one of these in the first part of today's lecture, which was protein disulfide isomerase. These form bonds within a single molecule, right? So PDI actually helps form the disulfides in the secretory pathway. We've seen polymerases, and these are basically involved in synthesizing DNA and RNA. And when we're looking at DNA and RNA polymerase, we'll learn a lot more about these. There's kinases like SARC, which we saw just a little while ago in the first part of the lecture. And these actually add a phosphate group to molecules. And protein kinases are really important in signaling. And the opposite of the kinases are the phosphatases, and these actually remove the phosphate group. We'll see some examples of those later on. Then there's oxidoreductases. These are basically involved in redox reactions, oxidases, reductases, dehydrogenases. And then there's the ATPases, and they hydrolyze ATP. And there's a whole bunch of different ATPases in the cell. So typically, and just a general rule for this course, I don't necessarily ask you guys to memorize anything in tables, right? I really want you guys to sort of get the feel of the big picture of how a cell works in this course. That's the important thing, with sort of a focus on the nitty-gritty details of specific cellular processes that make a cell function. And in this instance, I don't typically have you memorize stuff that's in a table, but you guys should be familiar with all of these different enzymes and what they do, right? So, you know, you will see polymerases on exams, right? What's the polymerase doing? How's it working? You will see kinases on exams and phosphatases and things like that. So it's just important that you guys understand what all these enzymes are and what they do. And many of them, it'll become second nature by the time this course ends and you're taking your final exam. Okay, so now on to enzyme kinetics. And I'm sure you guys have all had this in biochemistry, so I'm not going to spend a lot of time on it. But, you know, rates of reaction increase with the substrate concentration, assuming that the concentration of the enzyme sort of stays constant, right? And it eventually approaches Vmax, okay, the maximum rate that the enzyme can actually function at, okay? And one important characteristic is the Km, okay, and the Km is the substrate concentration where you reach 50% of the Vmax, okay, 0.5 Vmax is where the Km is. And once again, this isn't something I'm going to ask you guys to solve, the rates of reactions for certain things. It's just important that you understand it. And if you're taking biochemistry, you'll probably have to calculate things like that. Now, we talked about last time how enzymes actually lower the activation energy, right? They don't change the total energy of the reaction. They just basically lower the energy at the transition state. Okay, so in an uncatalyzed reaction, you'll have your substrate, and then your substrate in the transition, and then finally your product. And the enzyme will actually lower the transition state. So when you get the substrate, you get your enzyme substrate, and then the enzyme substrate in the transition, then your enzyme product, and then the product is released, right? So it's just lowering the activation energy for a catalyzed reaction. And so here's a great example of that, and this really sort of shows you how the active site of an enzyme plays into this and helps it. Okay, so this is lysozyme, and it's a really sort of classic catalytic molecule, right? It's something that, you know, I use in my lab routinely, and what it does is it breaks down the cell wall of bacteria. Okay, lysozyme is really prevalent in egg whites, and it's there to sort of protect the egg from getting any kind of bacterial infection. Okay, and what it does is bacteria have in their cell wall oligosaccharides, and so when you take the oligosaccharide S here with the enzyme lysozyme, it forms an enzyme substrate complex, and then you get an enzyme product, and then the product is released from the enzyme, and in this case it's broken the oligosaccharide. Okay, and so in this instance you get a little bit of a simple distortion that in the transition state, and that actually lowers the activation energy to hydrolyze the polysaccharide or the oligosaccharide in the bacterial cell wall. All right, and so here's the chemistry behind it. Here's your oligosaccharide. It's just a chain of sugars, and then when it's bound in the lysozyme enzyme, you have two amino acids that sort of form the catalytic site, glutamic acid number 35 and aspartic acid 52, and you get a nucleophilic attack on the C1 carbon of one of the sugars, and then that actually causes the proton to be removed from the glutamic acid. Then water comes in, and it basically associates with the sugar in a hydrolysis reaction. The water is consumed, and now you have an OH group on one, and then the proton actually goes to the other one, and that basically breaks the oligosaccharide and loosens the cell wall of the bacteria, and the bacteria eventually will lyse because of it. Okay, and hence the name lysozyme. All right, so this is just an example, and once again, don't get overly caught up in all of the details here. This is not a course in biochemistry or chemistry. It's a course in understanding enzyme, in cell biology, and to do that you have to sort of get a big picture of how enzymes can function, right, to lower activation energy. But there's also different models too, right? You can create a situation where an enzyme can bring in two different substrates and put them in close proximity to really create a favorable environment for the reaction to occur. You can also have active sites that change the sort of the electrostatic charge and basically causes sort of a rearrangement in the product to help catalyze the reaction. Or you can have sort of mechanical forces that help a reaction to occur. There's also cofactors that come into play in enzymes. We've seen a couple of these already. You have this molecule right here, which is called retinol, and that's contained in rhodopsin. And here is the structure of heme, which is present in hemoglobin, which is needed for carrying oxygen around. I know you guys can think of some others that are there. You can, great example is chlorophyll in the light harvesting complex in plants, which is involved in photosynthesis. But I'm sure if you think about it, you can find some more. Protein I work on in my lab, or at least I used to, actually had a flavonoadenine dinucleotide cofactor in it, and that actually absorbs a photon of light to give it transcriptional activity. So there's plenty of examples of these. These are just a couple here. You also have hemes in the electron transport chain involved in oxidative phosphorylation. So proteins can also act as catalytic tunnels, and in this instance, this example here, we have Carbomyol Phosphate synthetase, phosphate synthetase, and it actually has three active sites. There's active site 1, active site 2, and active site 3, and it creates this little tunnel here, and oftentimes molecules are sort of going to create these tunnels because the products of a various reaction can be quite reactive and cause problems. And so, these catalytic tunnels can exist to shield the products which may serve as a substrate in a downstream reaction from just reacting with the cell. And two examples of these are NRPS, which is non-ribosomal peptide synthetase, and polyketide synthetases that sort of create these little sort of reactive enzymes that shield the products from the rest of the cell as the products are being made. Okay, so if the intermediate is toxic or something, it's not uncommon to find these catalytic tunnels. So let's look at what Carbomyol phosphate synthetase does. Once again, nitty-gritty details. You guys don't have to memorize this, but I think it's important that you understand the big principle here. Okay, so here's carbomyl phosphate at the sort of the end. This is the molecule that's going to be made, and in active site 1, glutamine becomes glutamic acid, and that releases a molecule of ammonia. The ammonia can diffuse through a channel, and then in the second active site you have bicarbonate, and a molecule of ATP is consumed and converted to ADP, and the phosphate is actually added to the bicarbonate to generate carboxyl phosphatate. And you can see here, just by all of the net negative charges here, how reactive this could be to the cell and how sort of toxic it could be. But ultimately, ammonia comes in, and then you're left with carbamate, and then the carbamate, which is also reactive, can go on and become carbomyl phosphate by the addition of the phosphate group from ATP onto the end of it. So here you can see the phosphate. And so no need to memorize this. The important part is that oftentimes there's numerous intermediates in various biochemical pathways, and enzymes have oftentimes evolved to sort of create these sort of channels that the substrates can go through that keep them from reacting with other things in the cell. Enzymes also are oftentimes composed into large multi-subunit complexes. And the idea here is that you want to increase the rates of reaction. Okay, and so in this case, we're looking at pyruvate dehydrogenase, and it's actually composed of 8 trimers of lipoamide reductase transacetylase and dihydrolipol dehydrogenase, 12 molecules of that, and then 24 molecules of pyruvate decarboxylase. And these all come together basically to help convert pyruvate to acetyl coenzyme A. And that's the real goal of this whole complex. Okay, so enzymes can function or can be regulated by negative feedback. Okay, and negative feedback is one of these things that we'll see a lot over the course of the term. Okay, and it's always sort of drawn as a box here with a line. Okay, sort of almost as a stop sign. Okay, so in this instance you have A to B to C, but B can actually be shunted into X, Y, and Z, and once a lot of the Z product builds up, it actually can prevent the change from B to X, and so A to B will be shuttled. And there's lots of lots of instances within the cell where negative regulation helps things. And as a matter of fact, if we didn't have negative regulation in the cell, we probably never would have evolved, right? It would just take too much energy. So this is just a very common mode of regulation where the downstream products can actually prevent production of the product that's there, okay? And so the buildup of the products can actually inhibit their own synthesis. And that's, by definition, what negative feedback is. Here's some examples of negative feedback in amino acid synthesis. Okay, so if you go back to last lecture and you look at the big biochemistry slide that was there, there's all kinds of biochemical reactions in the synthesis of different amino acids. In this case, we've got lysine, methionine, isoleucine, and threonine. And a lot of these start off from aspartate, and through the various chemical reactions, there's inhibition. So lysine will inhibit its own synthesis in 2 different places. And if there's not enough lysine around, that actually helps drive formation of the lysine. But if there's plenty of lysine around, you don't want to be making more. Same thing with methionine. It will block, basically homoserine from entering the pathway and becoming methionine. Same thing over here. Bottom line is negative feedback is super common. We see it all the time. And so I have a question here, if you guys can think of any places where negative feedback occurs, and we'll see this throughout the course of the term. It actually occurs not only in biochemical processes, but it occurs in the cell cycle, it occurs in transcriptional regulation. There's a whole host of events that basically rely on negative feedback for control. You can also have positive feedback, okay? And here's just a great example of positive feedback where you have an enzyme that's actually inactive. And when you add in a new molecule, basically something that's going to bind outside the active site, that can actually make it active, okay? So without the, and this is called allosteric regulation, okay? So you have a molecule that can bind outside of the active site. And so here you have an enzyme with no positive regulator around, and it might be 10% active or something along those lines, okay? That's just an example. But when you add in the allosteric positive regulator, that will cause the enzyme to possibly change conformation and become much more active. And in this instance, it caused it to be 100% active. Okay, so positive feedback occurs by allosteric regulation, and in allosteric regulation, an enzyme has two binding sites. It's got an active site for the substrate over here, and then a site for the allosteric regulator to bind. And this can oftentimes change the conformation of the enzyme. Same thing works with allosteric inhibition or feedback and this can sometimes be how feedback inhibition works. Okay, so you have a molecule that's active, and then you can add in an allosteric inhibitor, which binds to a different portion of the protein and then makes it inactive. So without the molecule around, the allosteric inhibitor, the enzyme is 100% active. You add in the negative regulator of that, and then it can drop the activity of the enzyme quite dramatically. So you can have positive allosteric effects or negative allosteric effects. And this concept is actually really important for pharmaceutical development, and it's something that all the tox students that are in the course should really sort of have a good handle on. And I know by the time you guys graduate from graduate school, this will just be all second nature to you. Okay, another way proteins are regulated is by phosphorylation. Okay, and protein phosphorylation can have dramatic effects on protein activity, both positive and negative. And we're going to see this over and over again. It's a very huge reoccurring theme in this course, and I want you guys to really appreciate it. Okay, so if you have basically a molecule, a protein kinase is going to remove the gamma phosphate from ATP and actually add it to the molecule. In this case, it's covalently added to the serine side chain. Okay, and that's what the protein kinase does. Protein phosphatases will remove that phosphate. Okay, and so you can have things sort of in both directions, right? With the phosphate on, with the phosphate covalently attached to the protein, that can cause the protein to be on, or with a phosphate added to the protein, it can cause the protein to be shut down. Okay, and so kinases add the phosphate, phosphatases remove it, and one thing that will become sort of important later on is this phosphate can also, the addition of the phosphate can also lead to protein turnover. And I have a question here, does anyone know when this happens? And normally I would be asking you guys as a group. Hopefully someone in the class would raise their hand and say, oh, I think that happens during the cell cycle, and the answer would be correct. Okay. It also happens during transcriptional regulation. It happens in the circadian clock. There's a number of instances where it happens, where phosphorylation can lead to protein turnover. Here's just the structure of protein kinase domain. We actually saw this same domain in SARC, and so I encourage you guys all to look back at the image of SARC that I showed in part A of today's lecture, and you'll see that it's very similar. And the protein kinases are actually a huge family of proteins, and they all sort of phosphorylate different things. This is just, in this image, it just shows you these red arrows where amino acid differences occur in different kinases. Okay, but the thing is they all sort of share this kinase domain, and they all contain sort of a phosphate binding loop in them. All right, so I encourage you guys to all go back and look at SARC. Here's just some of the family of kinases. You have the tyrosine kinase subfamily. Here's a receptor serine kinase family. You got your cyclin-dependent kinases, which these are involved in cell cycle control, MAP kinases, and then there's a whole bunch of other ones. We'll see myosin light chain kinases later, calcium calmodulin dependent kinases. There's a whole bunch. No need to necessarily memorize this, but you guys will see a lot of these kinases throughout the course. And here's sort of how the phosphates can work on different proteins and how phosphorylation can change things. And this happens to be an example of cyclin-dependent kinases. And we'll cover this when we cover the cell cycle, but the idea is you'll get basically cellular inputs, right? And this can then ask a question, which, has this phosphate here been removed? Has this phosphate been added? And is cyclin present? Okay, and if the answer to all these questions is yes, okay, this phosphate doesn't exist, this phosphate is here, and you have cyclin present, that actually says that to turn on the CDK kinase activity, okay? And then that will go on and then phosphorylate other things in the cell cycle and lead to cell cycle progression. And once again, we'll see this. For now, all I want you guys to sort of do is sit back, take in that, you know, there's inputs that come into a protein via phosphorylation, and these can lead to outputs. Okay? And that becomes important in signal transduction, which we'll talk about later. And here's sort of what can happen in a signal transduction sort of pathway. Okay, so here you have your protein kinase, where you have an SH2 domain binding probably a phosphotyrosine here, and then you have your tyrosine here, and once this phosphate's removed, the SH2 domain no longer binds, and so it can undergo a conformational change and bind to a different ligand that actually is also phosphorylated, and then that can actually create and make it so the kinase becomes active and actually gets phosphorylated and basically becomes self-activated, right? So signal transduction is sort of a multi-step process, and there's also all kinds of signaling cascades. Another protein that's really important in signaling are G proteins, and G proteins are going to come up time and time again over the course of the term, or over this entire course. And so I really just want you guys to understand how they work, okay? It should be very straightforward, and I drive this point home, and I think I do a good job, and everybody leaves this course knowing exactly what G proteins do, okay? G proteins are also a large family of proteins, and when they're in their GTP-bound form, they're actually active, okay? Just like sometimes kinases, when they're phosphorylated, become active. G proteins, when they're bound to GDP, are active, okay? When you get GTP hydrolysis, it becomes inactive, and then eventually the GDP is exchanged and removed, and then you have an inactive enzyme, and it's replaced by GTP, and then it becomes active again. So G proteins have a huge important regulatory role in numerous cellular processes, in cell signaling. They're also involved in translational regulation. We'll see those see an example in a second. G proteins are also involved in vesicle formation in the secretory pathway, as well as vesicle fusion in the secretory pathway. So once again, G proteins, we'll see them over and over again, so you guys should really know how G proteins function. And here's an example of a prototypical G protein. It's RAS. So RAS is an interesting molecule. Many of you guys have probably heard about it. It's integral in cell signaling. There's the RAS MAP kinase cascade that I'm sure you guys have all learned at one point in time, but it really is the prototypical G protein. Here's the structure of it. When it's bound to GTP, you actually have this sort of switch helix that's sticking out, okay? When the GTP gets hydrolyzed to GDP, the switch helix can actually change its conformation, okay? And depending on how it is, whether or not it's bound to GDP or GTP will depend on the location of this switch helix and then determine its activity. Okay. Now, what controls the GDP and GTP are these enzymes called GEFs and GAPs. And we'll see these again over the course of the term, okay? A GEF is a GTP exchange factor, and a GAP is a GTP activating protein, all right? And we'll see these in a second and how they work. And we can sort of compare how G proteins work relative to phosphorylation in cell signaling, okay? So in the case with protein kinases and phosphorylation, you can get a signal in, and then all of a sudden the kinase will phosphorylate the protein, and then you will get a signal out, and then the protein phosphatase removes the phosphate, and it goes back to its resting state, okay? And this is basically how protein kinases and phosphorylation are involved in cell signaling. In the case with G proteins, you can get a signal in where you have your G protein bound in its GDP bound form. Once again, GPP here is GDP. And the GEF will exchange the GDP for GTP. And so when the G protein is bound with GTP, it's in its active form. Okay, and then the GAP protein, or the GTP activating protein, will remove the phosphate and send it back to its resting state. Okay, so this is something that, you know, we'll see these two things when we get to cell signaling quite a bit. This is one of those big picture items I really want you guys to understand. How do protein kinases and phosphorylation function relative to how G proteins and GEFs and GAPs work? Okay, so just know your GEFs, know your GEFs, know your GAPs, know your kinases, know your phosphatases, and have a sort of a real good understanding. There was something that I didn't mention on the last slide here with RAS. So it turns out that RAS is what's considered an oncogene, okay? And we'll look at oncogenes later on, but oncogenes are basically genes that can sort of give rise to cancers, okay, when they're mutated. And so in the case with RAS, there's mutations in RAS that actually will lock it in its GTP bound form, okay? So basically the protein is always active, and that leads to aberrant cell signaling within the cell, and so then the cancer cells just continually proliferate. Now it turns out that mutations in RAS are actually responsible for about 30 to 40% of all cancers. Okay, they don't have an exact number, but there's a range. I've seen both 30 to 40%. And so mutations in this is responsible for 40% of all cancers, or is found, not responsible, is found in 40% of all cancers. Okay, so here's an example of a G protein in action, and this happens during translational regulation. This is bacterial regulation, and the G protein is elongation factor thermo-unstable, or EFU2. Okay, and it plays a regulatory role in translation. Okay, it binds to an amino acyl charged tRNA. Okay, so here is your tRNA, and then you'll have your amino acid linked to the tRNA. Okay, and then you have EFU2 that sits out here, and it's a G protein, and it's normally bound in its GTP bound form. Okay, and so during translation, if there's proper base pairing between the tRNA and the mRNA, if you guys remember from last lecture, the codon and the anticodon, what will happen is the GTP here gets hydrolyzed by the G protein EFU2. And then this basically causes a conformational change in that switch helix, which releases the tRNA, which releases EFU2 from the tRNA, and then the amino acid can be added to the growing polypeptide by peptidyl synthetase. Here's another schematic of that, where you can see here's the ribbon structure of EFU2, here's GTP, this is the switch helix that I was just talking about. So it's in one conformation with GTP, and it's binding to the tRNA, and then if there's good base pairing between the codon and the anticodon, the GTP will be hydrolyzed to GDP, and then you get a conformational change in the switch helix, and you can get release of the tRNA. Okay, so that's basically how that works. Now these conformational changes with GTP, you can find sort of similar things that happen with ATP, and these actually occur within motor proteins, specifically the myosin motor proteins, right? And there's numerous different myosin proteins in a cell, and they all use ATP. And so what ends up happening is ATP binding causes an allosteric conformational change, and then that will actually, cause this to bind separately. The blue here is actually actin, and so then it'll bind, and then you get hydrolysis, and there's another conformational change, and then ultimately the ADP in the inorganic phosphate is released, and it goes back to its resting state. And I have the question here, where is this important? It's actually involved in muscle contraction for one, and there's basically cellular movement and all kinds of other things. And when we get to the cytoskeleton and motor proteins, we'll look at this closely. This is once again just another sort of big picture thing that we're going to really start to focus on when we move through the course. Okay, and in the case with muscle contraction and just with myosin motors in general, repeated cycles of this cause sort of movement in a direction. So once this happens over and over again, you get repeated cycles. So you can also have ATP binding proteins that are involved in transport, and a great class of these are called ABC transporters. And ABC transporters are great at moving small molecules across the lipid bilayer. So here you can see the lipid bilayer sort of shaded in gray. These are transmembrane spanning alpha helices, and so here you have a multi-subunit ABC transporter, and down here in the cytosol you can sort of see you have these ATP binding subunits. And so these, as they hydrolyze ATP to ADP, they actually undergo a conformational change. And these are actually conserved from bacteria all the way up to humans. And so bacterial ABC transporters, they function like this. They'll take in substrates from the outside, and then, it's in its resting state. And then when it binds two molecules of ATP, there's a conformational change, and then the substrate molecule can pass through, and then the ATP is hydrolyzed into ADP and inorganic phosphate, and it goes back to its resting state, and then the molecule can then enter into the cell. In the case with eukaryotic or mammalian ABC transporters, oftentimes they're involved in moving stuff out, and so you can have a small molecule in the cytosol. It'll sit sort of in this vestibule here, then two molecules of ATP will bind, and that causes a conformational, the ATP binding causes a conformational change, and then it's hydrolyzed, and the small solute can move into sort of the middle, and then the ATP is hydrolyzed into two ADPs and two inorganic phosphates, and it goes back to its resting conformation, and then the solute's free to basically move outside the cell. Okay, so this is just another example of ATP binding and hydrolysis that can cause conformational changes in proteins. Now, one of the things that gives cells some of their sort of really dynamic characteristics are that various subunits can be interchangeable. Okay, and if you want to look at it this way, the human genome's not as complex as we would like to believe. Okay, there's only about 20 to 25,000 proteins or so that do everything. And if you sort of take a step back now and think about a typical mammalian, a typical mammal, or a typical human, we have literally thousands of different cell types. Okay, but only 20 to 25,000 proteins that are doing everything. And the cells get around this by using different protein complexes to do kind of the same things on different substrates. Okay, and a perfect example of this is the skip colon protein. And this is involved in protein turnover, and we'll see this again when we cover protein turnover. But you have the colon protein here, which is basically just a large scaffold protein. And then you have skip, which is an adapter protein. And then attached to that is an F-box protein. And then you have your E2 ubiquitin conjugating enzyme. Okay? And so depending on what the F-box protein is, you can have a different target protein, and that will add ubiquitin onto a specific target protein. And then this protein will be targeted for degradation. And so there's many different F-box proteins and many different sort of adapter proteins that can actually function in this to basically target different proteins for destruction depending on when they're needed to be destroyed by the cell, right? So there's sort of one major mechanism to do this for all kinds of different proteins. And that's sort of shown here, where you have basically the colon protein, the scaffold protein, and skip, and these will sort of associate with all kinds of different F-box proteins and adapter proteins to bring in the ubiquitin conjugating enzymes. And then depending on which F-box protein is there, you can actually sort of generate turnover of all of these different enzymes, either the origin of replication complex, cell cycle regulators, the kinetochore, methionine synthesis, to help regulate what's going on in the cell. And so this is just a perfect way that a cell can use a limited amount of proteins to sort of do a wide array of functions, right? You don't need the same, you don't need a different skip colon every time you want to turn over a protein. You can have multiple different ones that sort of share subunits, right? And these lines just show that, you know, these have been shown to interact with different things. Subcellular localization is also really important. We'll see this sort of theme throughout the course, where you can have sort of a structure, a scaffold protein that can bring a whole bunch of proteins together to create a product. Okay, and scaffold proteins become really important, you know, in sort of subcellular localization. If you want to sort of think of you know, nerve cells, you'll have a dendrite and a nerve terminal, and they'll come together, and they're scaffold proteins that are basically holding neurotransmitter receptors in the right location within a cell. And this sort of, you know, image sort of shows how proteins are made, and then they can actually come together to form complexes. And this is just a immunofluorescent or fluorescent cell imaging to sort of show how localization becomes important. You can also get all kinds of covalent modifications on proteins, and in this instance here's P53. This is a really important tumor suppressor that we'll see throughout the course, and you can have a transactivation domain which gets a lot of phosphorylation and phosphates added to the N terminal of it, and then there's a DNA binding domain, and then you have other domains, a tetramerization domain and a C-terminal regulatory domain that get all kinds of other modifications. And we saw some of these modifications in the last lecture, like acetylation, phosphorylation. Sumylation is a little bit like ubiquitination, and it's actually a small ubiquitin-like molecule. And so here's just some examples of the modifications, the covalent modifications that can happen to P53. Covalent modifications also happen to on histones, and we'll look at this very closely when we move to transcriptional regulation, as well as looking at chromatin in general. Okay, so this is histone H3. This is just a portion of the tail, the N-terminal tail region of it, and you can see that, you know, this lysine at position 4 can be either methylated or acetylated. Likewise, this lysine at position 9 can be either methylated or acetylated, and these different modifications can have different effects on other proteins, okay? And they can be added and removed. And we'll look at this in much greater detail later. And here's just a chart of some of the different covalent modifications. You can get phosphates on serines, threonines, and tyrosines. Methylation usually occurs on lysine, as does acetylation. You can get a palmityl group on a cysteine, which can actually drive a protein. The palmityl group is just a big hydrophobic fatty acid side chain that gets added to cysteine, and that can drive a protein to bond, to sort of associate with the membrane. And then you can have an acetylglucosamine added to serine, threonines, and ubiquitin added to lysines. And a polyubiquitin chain can be used for protein degradation, and it can also be used for other things as well. Monoubiquitination versus polyubiquitination cause two different things, but we'll get to that later on in the term. And so that's all I really have for today. Read your assignment for chapter 4. I notice on this, this is probably for edition 5. I forgot to put of the book. I don't have the reading for edition 6, but it's going to be on your syllabus. And review the material I covered today in class. And as we get into the histones and ATP-dependent remodeling, a lot of the stuff that I covered today will be important when we get into cell signaling. We'll see more with G proteins and the kinases and all that kind of good stuff. So just remember, this lecture today and the lecture last week were basically review lectures and sort of also introductory lectures to tell you what we're going to be moving to as we move on. Don't get mired down in the nitty-gritty details of everything on every slide. Try to just take in the big picture of things, and what's important will become apparent as we sort of move on through the rest of the course. Okay, so I hopefully will see all of you guys this afternoon at 3:55, and I will talk to you then. Thanks.